everyone. My name is Rowan, and I'm a member of the Public Relations Department for the SCAA. Welcome back to our AEP session. If you haven't yet, make sure to click that like button and subscribe to our channel, as it will greatly help our platform move forward and help us spread awareness about the sickle cell disease to the general public. Today's session features medical student and researcher from UCSF, Alison. In today's session, Alison will be going over the two categories of hemoglobinopathies. He will be explaining his project and finding the relationship between the XMNL polymorphism and HbF, which is fetal hemoglobin. He will also be informing us of the genetic engineering of sickle cell anemia mutation by using CRISPR-Cas9. Finally, he will tell us about the importance of the public health perspective, such as informed consent, and how important it is that researchers are transparent. This is important to understand, as sickle cell patients are seen in a negative manner. It is important to understand the risks and gains from CRISPR-Cas9. Remember, you are a warrior, an inspiration, a motivator, and so much more. Without further ado, we welcome our guest, Alison.
All right, so I'm going to be talking about hemoglobinopathies is like this word, right? It's just blood diseases. So I'm going to be talking about blood diseases and kind of their categorizations. I'm going to be talking about the research that I'll be doing and the kind of newly discovered genetic engineering and clinical trials that is starting in sickle cell disease. Okay, so blood disorders, basically they're categorized into two things. So one categorization is that there are genes, right, that affect structure and quality of hemoglobin. So sickle cell disease, right, it's the quality of the hemoglobin. It's sickling of the hemoglobin. And another kind of blood disorder that there is are in genes that affect the quantity of hemoglobin. So thalassemia, right? Thalassemia is not about the quality, but it's not about the, that should be, yeah, so it's about the quantity. So you can't produce enough blood cells, okay? So hemoglobin variants, there's a lot, right? There's a lot of different diseases that are classified as sickle cell diseases. Sickle cell anemia is only one of them. There are thousands, right? Thousands and thousands. And they all have different clinical manifestations, and I'll kind of get into that a little bit later. And for thalassemias, there's alpha and beta thalassemia that is qualified under hemoglobins that are quantitative diseases. Okay, so sickle cell disease, kind of the basis of what it is. So we have hemoglobin A, okay? So hemoglobin A is basically, as adults, most of our blood is made up of hemoglobin A. So hemoglobin A is made from 4 proteins, okay? There's 2 alpha chains right here, you guys can see, alpha, alpha, beta, beta. And there's a heme group on it that carries the oxygen, okay? So this is basically what a red blood cell is made out of, 4 protein chains, okay? And so how sickle cell anemia, for example, manifests is kind of here. So we have the DNA sequence, right? This is referred to as the code of life. It's the instructions to make protein, right? So 99.9% of our DNA is all the same, right? But there's so little that's different, and those are the ones that can, cause disease. So sickle cell disease is basically a flip of a T. So in regular people, there's a T in the DNA sequence, and it's switched to an A. So this mutation, right, we call it, renders a different amino acid. So amino acids are basically, they basically read the genetic code. is basically what it is. Okay, so if there's an error in the genetic code, there's an error in reading the code. Okay, and you get defective protein, basically, right? So the defective protein. So the defective protein right here, you could see it affects the shape of the red blood cell, and you get a sickling of the cell versus your normal, red blood cells. They look plumpy, they can carry oxygen. Okay. So I just want to say that this sickle cell disease manifests in the beta chain gene. So remember we talked about the protein, the beta chains right here. So this gene defect causes a protein defect in the beta chain, right, which makes up the blood cell. So the cell changes shape because of the disease. Okay, and yeah, so this is what we start with, right? We start with DNA, and DNA is super, super long. It's like, if you watch videos, it's like the DNA in your cells, they go like from Earth to like the sun 100 times back and forth. It's crazy. They're just super, super, super long, codes of life that exist within us. Next slide. Okay. So as I said, right, there are many, many different mutations of, we call it, more inclusively sickle cell disease. The more, the deadlier ones are on the beta chain. Okay, so these are just some examples, right? So this is on the 6th amino acid. This is sickle cell disease. And then there's hemoglobin C, there's hemoglobin D, E, O. It's, as I said, it goes on, and every year they find new ones. And I just want to say that the more deadlier one is on the beta chain. There are also mutations that are on the alpha chain that makes up the red blood cell. So the alpha chain is kind of less predominant in disease because it makes it makes a minor part of the hemoglobin, right? So the majority of the hemoglobin is the beta chain. Okay, and then so thalassemia, thalassemia. So hemoglobin Hashron. So this is a mutation on the alpha chain. So I just wanted to kind of show an example of mutations on the alpha chain and the beta chain. And the beta chain is usually more deadly. Okay. So, okay, I just want to touch about thalassemia as well, because there's also research being done on thalassemia for genetic engineering. So I kind of wanted to introduce that as well. So as I said, thalassemia, so let's basically first start off, right, with that we get 2 pairs of genes, right, from mom and dad. So for each gene we have, so if you have the sickle cell trait, right, if you have one gene that's defective, you have the trait, because you have another gene that can produce enough hemoglobin, healthy hemoglobin for you. For thalassemia, right, so thalassemia is as I said, it's a quantitative disorder. So instead of there being a mutation, there's a deletion of a gene. So the more defective, as I said, in the thalassemia or in any sickle cell disease, is a defect in the beta chain, right? So the main clinical symptom is not having enough blood cells in thalassemia, and therefore you don't have much oxygenation of your tissues. Okay, and because you don't have enough blood cells, patients require blood transfusions, and it depends on what deletion is caused, right? So here's what we can talk about is beta thalassemia major. Okay, so beta thalassemia major is very, very severe form of thalassemia. It's when both of your genes, there's deletions on both of your genes. So you're not producing enough of that protein for your red blood cell. Okay, and then we have also beta thalassemia minor, and that is basically one of the genes deleted, and the other gene can produce enough red blood cells. So we have a little bit of less severe anemia compared to beta thalassemia major, okay? And then there is another disease that is sickle cell beta 0 thalassemia. So one gene is deleted, okay, and the other gene produces sickled red blood cells. So it's a qualitative and quantitative disorder, both kind of like a combination. And this is also usually patients go through a lot of blood transfusions, and it's also pretty severe as well. Okay, so basically I want to talk a little about the research that I did. It's about it's about hemoglobin, talking about fetal hemoglobin. I call it hemoglobin F, raising hemoglobin F in patients who have blood disorders and how it helps them clinically.